Good afternoon, everyone, and welcome to the ground rounds today. Hopefully that's not echoing too much. My name's Sarah Young. I'm head of the pathology department, and it's my great pleasure to introduce Professor Mike Eccles today. Mike is funded by the Cancer Research Trust as a leading thinker chair, so he's a research-only professor and has a significant amount of expertise in epigenetics and cancer. And he's going to be speaking today about some of this work that is funded through the Healthier Lives National Science Challenge on liquid biopsies, but also going to be talking about the concepts of tissue banking and some of the ways that we can facilitate that to enhance our research and the whole bench to bedside translational and research impact around diseases such as cancer. Thanks, Mike.
Great. Hopefully everyone can hear me all right. Great. Thanks, Sarah, for that introduction. I just put you on the spot a little bit there, but thank you very much. So I want to talk today about the idea of tissue and data banking, just to sort of get a feeling of what sort of interest there is and whether there's any sort of appetite for this sort of thing in the Deneen School of Medicine and the SDHB. So tissue banks have been obviously around for many years, long time, decades, and registries similarly have been around for a very long time. So the question is, the fact that these have been around, registries can actually exist on their own, independent of tissue banks, but quite often registries are also associated with tissue banks because they often contain codes that link information and specimens to their donors' identities. So a couple of very good examples of tissue banks in New Zealand include the Auckland Regional Tissue Bank, which is part of the Auckland District Health Board, ADHB, and also the Cancer Society Tissue Bank, which is at the Christchurch School of Medicine. But clearly these types of tissue banks can facilitate translational research. So the translational research activity at the Deneen School of Medicine is coordinated by Health Research South, which is a partnership between the DSM and the Southern District Health Board. And looking at what the Division of Health Sciences is involved in, it clearly is involved in a lot of research, including the translation of the research. And I'm going to be talking about a couple of these sorts of things today, including Genomics Aotearoa and the National Science Challenges, Healthier Lives. So New Zealand has a health research strategy through to 2027. And I'm not sure if you've actually had a look at this document. In general, it can be summarised under areas of common agreement involving the submissions that were made, a few areas of difference in the submissions. And there were some additional themes that were mentioned as well. Right down the bottom there, you can see the idea of building infrastructure. And I was reading this document and I was quite surprised that this idea of infrastructure came through time and time again in the submissions, which included the idea of building up national level data and tissue banks. So clearly a lot of people are thinking about this in New Zealand. But if you look at what global trends are happening in translational health research, I came across this review that was in Nature Reviews Drug Discovery. It was a little bit negative in saying that the development of new medical interventions simply takes too long, costs too much, and too often ends in failure. And also said that the decline in biopharmaceutical productivity, i.e. the innovation gap between fundamental discoveries and that there's a gap between this and the delivery of new therapeutic products. However, on the other side of the fence, I was at a conference recently at the New Zealand Society for Oncology, and there was a presenter by the name of Rohit Gupta, who's from the Stanford Biobank, and he was much more positive. He says that every sample has a story to tell, and biobanks and big data will change the world. So one of his more famous comments is that samples are not meant to live in a freezer. Their sole purpose in life is to produce data. And I was encouraged, as I've read further in this Nature Reviews article, that they were saying that the field of translational science is poised to provide solutions to these barriers that I just talked about, and to help bring more medicines to more patients more quickly. There's some good examples of this in Australia, in Melbourne and Sydney, at various research institutes. And also the UK has started on a very large sequencing project, which is called the 100,000 Genomes Project.
The 100,000 Genomes Project was announced by the UK Prime Minister in 2012 with the aim to better understand and treat rare diseases and cancers. With the project now well underway, Sue Saville visited Genomics England to see how genomics and personalised medicine will be a key part of NHS practice in the future.
Deciphering an individual's DNA is entering mainstream medicine in the NHS. Genomics England is setting up the infrastructure to tailor treatments to patients in the most advanced project of its kind in the world. The team running the 100,000 Genomes Project is making great strides. They've already decoded 14,000 full genetic maps or genomes.
We'll know how to choose treatments for cancer individualised to that patient's particular cancer. And
so, it's a very interesting video, goes on for some time. But
the 100.
Coming back to New Zealand, in the New Zealand research scene, what I want to do today is talk about an example of a project that we're doing that aims to incorporate genomic studies and collaborative tissue networking to improve outcomes for cancer patients. So in this work, what we're doing is we're taking blood samples from cancer patients, and we're taking the plasma from the blood samples, and we're isolating nucleic acids. And this, we should be able to answer several questions that relate to cancer prognosis, diagnosis, prediction, and recurrence. And this is part of the Healthier Lives National Science Challenge project that we're doing. And the work I'm going to talk about in the next few slides was actually done by Sandra Fitzgerald, who's in Chris Prince's lab at the Department of Molecular Medicine and Pathology in Auckland University. But there is this project is a multi-centre project, and there is another part of the project being done in Dunedin here in Professor Parry Guilford's lab, which is on plasma genomic biomarkers in bowel cancer. And in my lab, Ewan Roger, whose picture is here, has just started this project in Dunedin on melanoma. So currently the gold standard of, is histopathological analysis of melanoma samples or melanoma biopsies. So if a suspicious lesion is seen by a health professional, a biopsy is usually taken, and then that is examined, processed and examined by a pathologist who will look to see whether or not a melanoma lesion is present in the tissue sample. So what this project that I'm talking about hopes to be able to do is to provide a complementary type of technology which looks at plasma biomarkers, and this hopefully should be able to facilitate detection or confirmation of cancer, increased flexibility in the monitoring of tumour responses following treatment, and potentially for the earlier detection of tumour recurrence. So this, just a general outlining the general principles of what's involved in plasma, so-called circulating tumor DNA testing. One gets a blood sample from a patient, typically about 10 mils of blood, takes the plasma and isolates the nucleic acids. And then following this, tumors are shedding DNA into the blood at a fairly consistent rate. And if the tumor contains mutations that one can detect, such as, for example, shown here, the V600E mutation, then using various methodologies, you can actually detect that in the circulating tumor DNA in the blood. So this methodology has been around for a few years, and there are actually now already quite a number of publications which are rapidly increasing since about 2014. And so it was actually first found back in the 1960s and 70s that you could detect circulating free DNA in blood plasma. It was in 2012 that it was shown that minimal residual disease could be detected in blood plasma DNA. And since then, it's been looked at in a number of cancer types, including breast, colorectal, lung, prostate, pancreatic cancers, and so on. However, in ASCO this year, it was said that more evidence is needed to establish effective and appropriate use of liquid biopsy tests in people with cancer in the clinic. So there is a need for more work to be done on this. So melanoma, it turns out, is probably a perfect fit for doing liquid biopsy type studies because the mutations that occur in the BRAF and the NRAS oncogenes are very frequent. They occur probably up to about 60% of all cutaneous melanomas have one of these types of mutations. And some of our colleagues at the Edith Cowan University in Perth have, that's Elin Gray and Mel Zyman, have written this little review, and this is a table from that review that indicates that quite a number of studies have been published on melanoma liquid biopsy already. And so what we're currently looking at with Sandra Fitzgerald and my group here at Otago, we're looking at how sensitive this technology is. So as with all technologies, there will be a limit of detection. We can get down to around 1 mutant copy of DNA in about 1,000 wild types, or even as far down as 1 mutant copy in about 10,000 wild type copies. And so this is most achievable using Droplet digital PCR, which I'll talk about more in my talk today, detects single mutations in the plasma DNA. There are other technologies which we're also trying out, one called UltraSeq, which is a mass spectrometry type of detection method, and it's able to detect, it's about the same sensitivity as Droplet digital PCR, and it can detect multiple mutations simultaneously in the plasma. And then another technology we were trying is next generation sequencing, where one can detect even a couple of hundred mutations simultaneously, but it's a little less sensitive. So there are some variations in how these various methodologies compare. So the UltraSeq requires quite a bit more volume input in terms of DNA. I mentioned that about the sensitivities. Probably the lowest cost is the Droplet digital PCR, and also it has the lowest time taken for going from sample to result. So Droplet digital PCR seems like it's quite a good methodology for this type of analysis. So what actually Droplet digital PCR involves is the encapsulation of DNA into little droplets of oil. And so the less than or equivalent to 1 copy of DNA per droplet is encapsulated And along with that is all the reagents required for carrying out PCR reactions. And so as a result of doing the PCR, one can detect either the wild type or the mutant copies. So each of these spots or dots here represents one droplet, and that's the fluorescence of the droplet is detected as being either wild type or mutant. So to date, Sandra in Auckland has actually analysed 24 patient samples using all three, well, 24 of them have been analysed using Droplet digital PCR for mutations in these couple of genes. 21 have been screened with the UltraSeq method and 17 using next generation sequencing. All of these patients have been recruited through the Auckland Regional Tissue Bank, which has been fantastic for them to be able to do that. And what I'm thinking about now is, just as we're getting started in Dunedin, we'd like to really set up a Dunedin-based biobank for this kind of work. So I want to talk, just introduce or show you briefly some of the results that that Sandra's been getting with some of these patients. So on this slide is showing this red line here is the lactate dehydrogenase or LDH level, and that grey line is probably the upper limit of the normal level that you might find in circulating in blood. So in this patient who is a stage 4 melanoma patient who's actually on immunotherapy treatment, you can see from day 1 to about day 148, the levels of the LDH have been going up. And you can also see that the levels of the, shown here, the circulating tumour DNA shown in the blue columns has also been going up, which parallels the increase in the LDH levels fairly well. This patient also has had CT scans, and shown here is this stage 4 patient had a mass that's shown here in the groin of the patient, and you can see between the scan at before day 1 and a later scan at about day 100, this mass has actually grown in size. So this patient appears to be progressing while on immunotherapy treatment, and that's actually shown by all of these 3 methodologies that I've just talked about. The next patient here, this is a patient, also a stage 4 melanoma patient who is receiving immunotherapy, and here the LDH is sort of fairly flat. So, but the circulating tumour DNA showed it went up initially at day 41, and then it's come down by about day 80, and then it went up again since day 80, or since about day 100, and it's gone way up again. So what happened to the tumour? Well, this tumour did show at day 100 or so a sort of a reduction in size, which corresponds with the circulating tumour DNA result. But we haven't got a CT scan since then, and it appears from the ctDNA work that this patient may be relapsing, but we haven't actually communicated these results back to the clinician at this stage. The next patient, patient 3, you can see from the LDH that this tumour seems to be growing and progressing. You can see from the CT scan, this is nodules that are present, and you can see many more nodules in the lung at the CT scan at day 64, I think it is. And along with that, you can see that the circulating tumour DNA levels are actually increasing inexorably over this time frame. So this patient is also progressing on treatment with immunotherapy. And lastly, this patient, an NRAS mutation that was detected in this patient, This LDH also appears to be fairly flat, and it's under the levels of normal levels of LDH in the blood. You can see that these ctDNA, circulating tumor DNA, decreased over that time and sort of went away entirely. And on CT scan, this is a tumor mass, which is the first scan is under the right armpit here, a bit of tumor mass there, and the second scan is also under the armpit in a different plane. And although there seems to be some sort of mixed response going on in the CT scan, the CT circulating tumor DNA might indicate that this patient is having a response. Just to sort of summarize this, we're doing a lot more, and I've only talked about a few patients here. Sandra's got results on a lot more patients, and I'm not presenting here, but one of the patients, we saw that the circulating tumor DNA rose very significantly several weeks before we could see a rise in the LDH and also any changes that were happening on the CT scan. So this type of work suggests that it could be useful for the identification and monitoring of melanoma patients for treatment resistant or response or relapse. It also could be useful for patients who live in smaller centres in New Zealand who could send their blood samples for testing at regular intervals. So it's possible that Droplet digital PCR could predict response to treatment to immunotherapy, for example, and could be more accurate than LDH. It's possible that monitoring the circulating tumor DNA could complement standard of care treatment. But, as the ASCO thing said, we need a lot more work, and so this is really still under investigation. So we're going to continue collecting and monitoring melanoma patients' circulating tumor DNA. We're going to try and develop more sensitive assays. We want to integrate the clinical side and the genomics and the patient registries and patient electronic medical health records together. We want to also do a tube trial for those patients who are living in the smaller centres and see whether or not the DNA survives the several days that it takes to get from those centres into a testing centre and a more main centre. Collaborations are important, and also we want to, we have to move quickly. This research is spanning between translational research and the clinical realm. So we also think that this could actually shift some of the cancer surveillance potentially away from hospitals and closer to primary care situations. It might complement the use of CT and PET scans, and it could have implications for treatment decisions, such as whether to do adjuvant treatment or whether to continue on systemic treatment. And we hope that it will be specific for New Zealand patients and that it would be available eventually at an affordable cost. So what we need is, we need coordination between the clinicians and the scientists for this, and we need to develop our systems for data and tissues. This is going to require collaborations, and we also need to do further consultation with Maori and Pacifica experts in order to be able to reduce the inequalities that there are in the population. Not perhaps so much with melanoma, but certainly we're going to expand this hopefully to other tumour types like breast cancer, and it's already being investigated in bowel cancer in Perry's lab. The genomics infrastructure is probably not at the level, and it's a bit limited to be able to do the sorts of research that we want to do in New Zealand. So we need to develop better infrastructure, and one of the new approaches that is just to come around is the Genomics Aotearoa, who have just started their whole operation very recently, and it's been led out of the University of Otago by Professor Peter Dearden in the biochemistry department. And it's hoped that NESI, which is the New Zealand e-science infrastructure will help to host some of the systems that Genomics Aotearoa will develop. So Genomics Aotearoa will have several exemplar type projects. One of these will be the sequencing of 1,000 genomes of Maori individuals from around New Zealand. They also want to develop bioinformatics capability in New Zealand, and also the sorts of things that I've been talking about, which is the computational interfaces which might be available on the NESI systems. So in relation to this, I've been thinking for quite a while, and we've started some work on a potential data infrastructure called Cancer Hub, which is actually not part of GA at this point, but you can actually have a look at Cancer Hub. It's on www.cancerhub.net. What we're aiming to do is to allow researchers to be able to access New Zealand cancer patient data sets via Cancer Hub, and to be able to use online tools for doing cancer research, including hosting New Zealand cancer patient data in a repository, and having facilities like CBio portal to be able to carry out genomics analysis. We also hope that this portal and this facility will be able to support the involvement of patients in becoming involved in cancer research. So we want to be able to provide infrastructure for patients to be able to know what kind of things they need to do to be able to get involved in research. And included in this is consultation with Maori to actually do things which are appropriate for Maori to help to address the inequities that are in the population. And patients may be able to go onto the portal right now even, and from an amalgamation of different clinical trial sites that there are, we've put these together as a facility that patients can look for and find clinical trials on cancer in New Zealand. And also we're hoping to be able to develop a network, so researchers in Dunedin, for example, could look in Auckland and try and find a person who has similar interests, for example, who they may not know at this stage. So about 10 days ago, I had set up this online invitation for people to have their say. And so far, actually, we've had something like 27 responses, and I did my completion at 23 responses at 8 p.m. on the 3rd of September. But as they're still coming in right at the moment, and so 22 people gave their scores from 1 to 5 on 7 discussion points that I raised. And 11 of these also gave some additional free text comments. So I thought I would just share these with you and go through some of these discussion points. So discussion point 1 is that local integrated databases aimed at collecting and storing research data which are linkable to other health data, if done in the right way, will be an asset to New Zealand. And I was reasonably surprised that a lot of people actually fairly strongly agreed with this sort of statement, with a median and average of 5. One person said that they felt that integrated data is extremely important for innovative research, and the major barriers to the use of data reside at the approvals level. You can actually have a look at all of these points later on, and you can come back to this by going online, I think, to the talk. So I won't go through all of these individual points, but I'm just raising some things that have popped out that I thought were interesting. Discussion point 2 is the existing levels of protection are not adequate to protect patient privacy in the context of either genomics or clinical trials health research databases in New Zealand. And there was a fairly mixed response. Some strongly agreed, some strongly disagreed, but it was very mixed. Some people said they were very concerned as to who retains ownership of biobanked materials. One person said, what we need are sensible, informed discussions around how to best protect people's data, use these data in an ethical way, engage properly at all levels with Maori and Pacifica, and ensure these data are used for the public good. Discussion point 3 was that enhancing local collaborations and increased use of tissues and data will be important to facilitate more translational health research than is currently happening. So one person commented that a national database would be an excellent step in the right direction so that researchers can at least assess what tissues might be available. I think that they're talking about like having some kind of framework where you can actually see what's available across all the different tissue banks in New Zealand, which I think would be a great idea. So discussion point 4 was that lack of time and other resources at the disposal of healthcare professionals within the DHBs is impeding growth of translational health research capacity locally. And there was reasonably strong agreement with this statement. One person said that maybe the key is to start small and rather than to build all aspects of the system. Another person said that collaborative banking in New Zealand makes sense as our institutions are small. Discussion point 5 was that participation in or collaboration in translational health research, e.g. such as clinical trials research, is not really part of the culture of New Zealanders at the present time. One person said, what cultural considerations would be needed if biobanking was to be implemented more frequently in New Zealand? Another person said, collaborative banking in New Zealand is absolutely necessary to enable any meaningful research to occur here on low incidence type pathology, for example, pancreatic malignancies. Overall, that was a fairly mixed response to that statement. The discussion point 6 was that greater development of the concept of DHB-based translational research centres will enhance the growth of translational health research locally. And there was fairly strong agreement with this statement. One person said, I think this depends upon a collaboration between universities and DHBs. And lastly, discussion point 7 was that DHBs should be funded by the government so they can support infrastructure and resources for translational health research. And I was fairly surprised by the response to this. A lot of people actually strongly agreed with this statement. One person said, maybe a better option may be to separate out the funding like the UK does to the National Institutes of Health Research, which sits separate to individual NHS trusts. So I was, fairly encouraged by the responses, and I feel as though we should make an effort. And I want to also point out that the University of Otago has a strong foundation for collaborative tissue networking and being able to do associated data registries in a host of related areas. For example, we've got excellent tissue banking going on at the Cancer Society Tissue Bank in Christchurch. We have researchers here who are leaders in ethical and legal aspects of tissue banking and patient registries. And the Dunedin School of Medicine has strongly established and highly integrated links to the Southern District Health Board and to the Dunedin Hospital. The University of Otago and the Dunedin School of Medicine have strong reputations in clinical trials research. And so the Health Research South is also extremely good at the DSM. We have strong genomics capabilities as well as being the host for Genomics Aotearoa. As well as that, we have strong genetics research and teaching at Otago, for example, Genetics Otago. So all of these, I think, bode well for the idea of actually going forward and doing some more in regards to collaborative tissue networking and collaborative data registries. So what I'm thinking about is how to plan this and how to involve this in the research that I'm doing, and planning how to do next steps. So I think what we need maybe is to form a planning group here in Otago, and we need more consultation with Maori and Pacifica. We need consultations with our academic and clinical colleagues, many of whom are collecting tissues already. We need to develop the ethical applications for this. We need to think about the infrastructure that's needed for coordinating data and tissue repositories, and we need to develop a governance and an oversight of this activity. We need to think about how we can actually facilitate the staffing needs and gain some financial support. One thing I want to really emphasise is we're on the cusp of doing some major genomic sequencing with Genomics Aotearoa. And in particular, I think this all needs to be coordinated with Dunedin Hospital rebuild. So hopefully I've left some time for some discussion at the end, but I want to acknowledge my colleagues that are involved in this, and at the University of Otago, particularly Harry Guilford and Rob Day, Ewan Roger in my lab, Tania Slatter, Heather Cunliffe, and Sarah Gallagher has helped me put this presentation together, and Becky Laurie has been very important in putting this together as well. A lot of the stuff that I talked about was actually done by Chris Prince's lab group, particularly by Sandra Fitzgerald. But here in Dunedin, we've got collaborations that are coming underway with Chris Jackson, Debbie Skinner, Will McMillan, and Patrick Lyle and Anne Collins, and other collaborations up in Auckland as well. And also this has involved, as I said, the Auckland Regional Tissue Bank team. Hopefully we can develop a team here at Otago for doing this work. So I'd like to thank you all for coming along and listening, and any discussion will be welcome. Thank you.
Thanks, Mike. That was a great presentation. So Mike's happy to take questions now for the next 10 minutes or so before we have to move out for a class. So has anyone got any questions they'd like to ask or comments they'd like to make about some of these new initiatives that we want to get up and running here?
Want to use the...
Oh, yes, we need to pass the microphone around because quite a number of people watch this on live stream. In fact, a large number do, more than normally that attend. So we need to use the microphone, sorry, so you can throw it around like a netball.
Hi, Mike. Thanks for your presentation. It's really cool to see these ideas taking shape. Ourselves at the Faculty of Dentistry have been discussing similar things as well, doing a tissue bank, more dental-based one. So it'll be interesting to discuss further this with you guys. I'd like to know, how do you see this complementing or even overlapping with the work that's done with the Cancer Society Tissue Bank, and how do you see alternatives for funding for this project? Because my understanding is that the Cancer Society Tissue Bank, they use the university facilities and space, but the funding is provided pretty much 100% by the Cancer Society. So how could we do this happen here?
That's true. So I think that we would set up systems which are very complementary to the Cancer Society Tissue Bank and try to work with them as partners. And they have a separate board, so I don't think we would actually be under their governance or anything like that, but I think we would try to actually have systems which can interact with the systems that they have. So they receive, as you say, some funding from the Cancer Society in Canterbury and the West Coast. Well, maybe we could actually approach the Southland and Otago Cancer Society here to see if they're interested in providing some support along these lines. Maybe the Freemasons might be an idea. I think the Freemasons actually had provided support for the Auckland Regional Tissue Bank at the start. I think, obviously, the Cancer Society Tissue Bank in Christchurch gets a little bit of its revenue through charging for use of the sample. So there is that possibility to be able to charge, but although that's not going to cover the majority of the costs, I don't think. But I think the more we think about it, the more possibilities and things we will come up with that could facilitate funding opportunities for this.
Thanks for a really good talk. I was also thinking about funding and nationalised databases and that sort of thing. But if you were going to start within a local area like Dunedin, one way of generating some funding, if we just wanted to leave it to departments to actually do the banking of their tissue in the way they wanted, but have centralised IT. The benefit of having centralised IT is that reduces some costs and also enables us to have nationalised IT if we follow whatever Christchurch is using. It also helps to ease that ethics process, which I'm sure you've considered, and perhaps also ease that process of universal consent to help ensure that tissue that's banked and doesn't all get used can then be used for other purposes. But a way of achieving funding for that might be to create a bit of departmental FOMO. And then there's a lot of departments probably represented in this room that together would be able to generate a reasonable source of funding just for IT if that was all we were trying to achieve here.
I agree. So another possibility is if we could make it a research infrastructure that could be supported through the kind of the DRIG mechanisms and the Division of Health Sciences perhaps. But I agree with your points, and I think that to some extent, if we were able to tap into individual research grants with just a little contribution from each, then that could actually go quite a long way from different departments. Heather.
Hi, Mike. Thanks very much for your presentation. Just quickly, some of the things that we haven't talked on is the quality of the specimens. I know that Carolyn Compton, the director of the biospecimen effort at the National Cancer Institute for the Cancer Genome Atlas project, learned that, for the samples that they need to acquire from multiple laboratories, the science can fall over if the preservation isn't by a national standard. I know that there's a lot of guidelines, such as the ISBRA guidelines, that are now in place, and I think that that's something that if we're going to kick off an effort like this, perhaps we should suggest some national policy guidelines to flesh out that everyone should follow.
Yeah, thanks for that, Heather. I think that's a very good point. And I think that Christchurch has been setting up some pretty good systems for getting good quality samples. And it'd be interesting to actually sort of compare and contrast with what Auckland has been setting up and pick the best of both worlds, I guess, and set up and actually, I think, have standardized protocols that we know what kind of samples we're getting for what kind of purposes in the end, and that everything is as best quality as we can get. Yeah, thanks.
What about the question of focus? How do you, when you're setting this up, obviously we've each got our interests and we could itemize those. Is that where we should be starting?
Say that again.
Focus.
Yeah.
Because we can't collect everything, especially when that kind of information is really quite time consuming.
Yeah. I think that we should be pretty focused initially on projects that are at hand and what's actually required to make those projects work. So in the case of the one that I was just talking about, I guess we could focus on the requirements for the blood samples and try and get infrastructure that makes that work. If other people are bringing other projects, perhaps we need to make a decision as to what projects we are willing to bring in at this stage, just so that it's not overwhelming, but actually try and set up the requirements for each project so that we can make each project work, and not too many maybe at the start.
Thank you for the talk, very interesting. Do you think we have adequate computational support at Otago for this kind of initiative, or is this something that will need to be developed? And when I say computational support, I mean not just the hardware and things, but also methods and software. And,
I think we have very good capabilities. I've got a good team of bioinformaticians that I'm working with, and I think that computationally we can set up systems which can manage to do these sorts of things that I'm talking about, like bringing in CBio portal, making the appropriate repositories. At the moment, I am in discussions with NESI, and we're working, because they're actually just refreshing their systems and coming up with new infrastructure. And so I'm actually working with them to see if they can actually provide some support as well, that we can do something that actually is useful for more people than just our lab. Thanks.
thank you.
We might finish up there. Please, if you have any additional comments or questions, come and talk to Mike at the end. As Mike has alluded to, he's going to be setting up a group to discuss the idea about biobanking and how we could move forward. So if any of you have any particular interest in that, please contact Mike and make sure that you're known to him, so we can make sure that we capture your thoughts about that. Also, a couple of additional points. So as mentioned, DRIG, so the Divisional Research Infrastructure Governance Group, could be an opportunity, and we have discussed this in the past around funding, setting up some seed funding for a biobank. And also with the new hospital that's being built, certainly biobanking is part of the schedule of activities that have been captured by the planning team to go into a research area that's associated with clinical research in the new hospital. So we've got some great opportunities coming up in this space. So thank you very much for coming along, and just join with me one more time in thanking Mike.